Hey everyone, Azim here. This is chapter 14, slide 32. Chapter 14, slide 32. We're wrapping up our discussion on nervous cells and tissues. We've talked about the basics of sensory neurons, interneurons, and motor neurons, motor efferent neurons. Now we're going to put this all together by talking about how are these axons in the peripheral nervous system bundled together, and how are reflex pathways formed, or what are the components of a reflex pathway. Starting with this idea of nerves. A neuron and nerve, those are two different things. A neuron, let me draw it out here in pink. A neuron has dendrites, has a cell body, has an axon terminal. So I've drawn one neuron here. This is one example. I remember there's different neuron shapes, but here's one example of a neuron. I could draw another neuron next to it and another neuron next to it. These are axons. When neurons are next to each other, which they typically are, the axons bundle together. So all of these axons bundle together, and they go in a similar pathway. Just like if I drive my car, I could go off-roading. My car's not really equipped for that. I could just go off-roads, but I usually join a road where other cars are traveling. We're all traveling in the same pathway. Axons bundle together in similar pathways. These bundles, those are called nerves. Nerves are bundles of axons specifically in the peripheral nervous system. We'll talk about how axons come together in the central nervous system later. Nerves are bundles of axons in the peripheral nervous system. And they're organized and protected by connective tissue. So let me clear my drawing here. Shown here is a single axon of a neuron. What kind of neuron? Who knows? I don't know what kind of nerve this is. Depends where if we're coming or going. Actually, nerves are mixed, but we'll get to that later. But here's a single axon, and so there's a bunch of axons bundled together here. Surrounding and protecting a single axon is connective tissue, and that connective tissue is called endoneurium. Endo, because it's towards the inside of this nerve. Neurium is referring to nervous tissue. Endoneurium, the connective tissue that surrounds a single axon, even if it's myelinated. So it would be around the myelin. In this histology over here, it's kind of zoomed out, but all of these little dots here, these little areas, these are axons surrounded by endoneurium. Remember endomesium? It's the same pattern here. Same pattern as what we saw in muscles. Axons are bundled together into fascicles. So what I'm coloring here in green, and what I'm going to color here in green in this histology, this is a fascicle. And the connective tissue that surrounds a fascicle is perineurium. Perineurium surrounds a single fascicle. Then you bundle the bundles together, bundle the fascicles together, covering and protecting the entire nerve. That's epineurium. Epineurium is the outer layer. I know you might not know what dura mater is yet. We'll get to that later, but keep that in mind. The epineurium is going to be continuous with something called the dura mater, the outer protective layer of the spinal cord and brain. So, and that would be in our picture, that would be this tissue here. Endoneurium around the single axons, perineurium around the fascicles, epineurium around the bundles of fascicles, around the whole thing, around the whole nerve. And you only find this in the peripheral nervous system, not in the spinal cord, not in the brain. All right, let me actually show you where that is right here. In this picture here, what we have is, this is the spinal cord. So that's central nervous system. Let me draw the outline of it. Great. Okay, so there's our spinal cord, which is part of the central nervous system. If you look at these, it's off the page, but if you look at this extension here on one side and this extension here on the other side, this is a nerve. There's axons that go back and forth, sensory and motor, on either side. Spinal nerves exit on either side. And looking back to when we learned about bones, remember those intervertebral foramina, intervertebral canals? Nerves pass through intervertebral foramina on either side between the vertebrae. That's what a nerve is. A reflex, a nervous system reflex, is an immediate, involuntary response, and it does not always involve the brain. It does involve the brain or spinal cord. It has to involve some part of the central nervous system. It involves the brain or spinal cord. There are two different reflex types in terms of function. It can be a motor reflex. So you're controlling skeletal muscles like that knee-jerk reflex. That's a motor reflex. Or it could be an autonomic reflex, controlling your heart, controlling your blood vessels, controlling your digestion, controlling salivation, controlling other glands. That's an autonomic reflex. A reflex arc begins with a sensory receptor, ends at a peripheral effector. So we start with a sensory receptor, come to the brain or spinal cord, output and control something, control our effector. Let me erase this and then point out different things that are going on here. All right, step one, activation of a sensory receptor. That would be right here. So we have the dendrites of our sensory receptor here in blue. Our sensory neuron is here in blue. You can see that it's pseudo-unipolar. You can see that its dendrites are off somewhere in the periphery, but its axon terminal is in the spinal cord. It's in the central nervous system. That's step one. We activate this neuron and it sends information to the spinal cord so that we can integrate. Step two, we relay through this afferent pathway. So we're passing information along this axon. Step three, we process within the central nervous system. So that means with our interneuron, which is multipolar in shape, we're processing something. We're deciding, do we act on this information or do we not? Step four, if we do act on it, we send information out our motor neuron. Note how the axons are also in this nerve. Step five, we have some kind of response. If we're controlling a muscle, our muscle moves. If we're controlling an organ, something happens to that organ. We start digesting, we start secreting, whatever. That is a basic reflex arc. Sensation, input, integration, deciding, output, control. Sensory input, integrating, deciding, output, control. Let's look at an example. We saw one already, the knee-jerk reflex. Let's look at another. Here's the withdrawal reflex. With the withdrawal reflex, let's say you touch something hot, like this candle. When you touch something hot, you'll reflexively pull your hand away. So you've sensed pain because it's too hot, and you reflexively control your muscle to move your hand away. So step one, you sense something through that sensory neuron. That sensory neuron sends along this afferent pathway, we send it to our spinal cord in this case. So I guess that's step two. We're sending information to the spinal cord. Step three, we integrate that information in our interneuron. Step four, we send that information. We send a signal to our muscle to move our muscle because we want to move our muscle out of the way. Step five, we control our muscle, contract, move our hand away. That's one example of a reflex pathway. This would be a motor reflex, not an autonomic one, because we're controlling skeletal muscle. The knee-jerk reflex is also a motor reflex. If we look at this knee-jerk reflex, which is also a motor reflex, should say patellar ligament. So you tap the patellar ligament, and step one, your muscle spindle senses that stretch. Step two, your sensory neuron sends that information into the spinal cord. Step three, your spinal cord integrates that information. Do we do something about this? Do we not? We do act on it. So step four, we control our motor neuron and send that information to our muscle. Step five, we control our muscle. We kick our leg out. I think it'll be useful to see more examples. So I'm going to switch over to my whiteboard and draw some more examples. What I've already written out here are all the different things that we've discussed. I haven't drawn anything yet, but there's clearly a lot. We've gone over many different somatosensory receptors. We've gone over visceral sensory receptors. I have 2 pages here. And we've gone over special sensory receptors. Let's draw out each one how I'm asking you to draw them. So there's a very particular way that I want you to draw your neurons. So for drawing a Merkel cell, this is actually going to involve a special neuroepithelial cell. So I'm going to first draw a neuroepithelial cell that's found in stratum basale. And this talks to it's going to be a pseudo-unipolar neuron. Let me redraw how I drew my cell body here. All right, so there's our Merkel cell talking to the dendrites of a pseudo-unipolar neuron. The cell body is in a ganglion of some kind. We'll further describe what a ganglion is. It's a region of cell bodies of a neuron found in the peripheral nervous system. But I drew this wrong. The axon should extend into the central nervous system. And so I'm going to draw my axon terminal here in the central nervous system. A Meissner's corpuscle, it's what surrounds the dendrites. Meissner's corpuscle is connective tissue that surrounds the dendrites in the papillary layer. Pacinian corpuscle, also connective tissue that surrounds dendrites. Ruffini corpuscle, connective tissue that surrounds dendrites. Root hair plexus is just bare dendrites. It's free nerve ending. So I'm going to leave the dendrites alone. Nociceptor is also a free nerve ending. I'm going to leave the dendrites alone. Let me just make sure we're on the same page here. This is the cell body. These are dendrites. This is the axon. This is the axon terminal. Thermoreceptor is also bare dendrites. So I draw this the same way. Muscle spindles actually had a connective tissue covering. Whoops. So let's draw connective tissue covering around the dendrites. Same with Golgi tendon organ. That was also connective tissue covering around dendrites. For all of these somatosensory neurons, they're all pseudounipolar. Dendrites, cell bodies are in the peripheral nervous system. Axons, axon terminals have extended into the central nervous system. Let's just pause here for a second. I'll show more sensory ones, but for any of these somatosensory receptors, we can send this information to the spinal cord or to the brain. So I'm going to draw a neuron here, and it's going to be multipolar. So this is a multipolar interneuron. It's a multipolar interneuron. The dendrites surround the cell body. The axon exits potentially to the peripheral nervous system. Let me draw another one here. Let's say in response to feeling deep pressure, we decide that we want to move our arm away. So then we'd use our somatic motor nervous system. So now we're in motor output. These are also multipolar. So I would draw many dendrites around the cell body, and I'd control a muscle. This is my poor drawing of a muscle, which is our effector. So that could be a reflex pathway, what I've drawn here. You've got a sensory neuron, interneuron, motor neuron. A very simple one that I've drawn, because there's only 3 neurons. Reflexes can be complex. And we're going to stay simple here with like up to 3 in a pathway, maybe 4. But here's an example of how a reflex could happen. Let's go to the next page. I'm going to show you some more sensory neurons. If we have a visceral sensory, this is also pseudounipolar. Let's say we're detecting carbon dioxide levels from the aorta, so we're using a chemoreceptor. It's bare dendrites, cell body off to the side in a ganglion, and extends into the central nervous system. From there, that would send information to a motor neuron, excuse me, an interneuron. Which happens to be multipolar in shape. So it has many dendrites. Let's say we detected low levels of carbon dioxide in our blood. Let's say if we're exercising, let's say we detect high levels. Let's say we detect high levels of carbon dioxide in our blood. If we're exercising, we're building up carbon dioxide levels in our blood. So what do we need to do? We need to breathe harder and faster to get more oxygen in, to get rid of carbon dioxide. So we would want to stimulate our sympathetic nervous system, another branch of the afferent division. A sympathetic pathway, we would have, oh, you know what? I've drawn this wrong. I'm going to correct what I did earlier. But a sympathetic pathway Interneurons are entirely within the central nervous system, so that's where I went wrong. So there I fixed where the axon terminal is. Sympathetic pathway starts in the central nervous system, and then it extends out. Let me use a different color. And we're actually going to learn that there's a second neuron in this pathway. Let's say we control the heart. There's a ganglion here. So with the sympathetic pathway, we have motor neurons. These are multipolar. These are multipolar neurons that extend to the heart. Say we want to speed up our heart rate. That's one example of using a reflex. This would be a visceral reflex because we're controlling the gland or the heart. Another visceral reflex, say we need to pee, say we need to urinate. We can detect the stretch of our urinary bladder with a pseudounipolar visceral sensory neuron. Process that with an interneuron in the brain or spinal cord, probably the brain. And then using our parasympathetic nervous system, we can release. There's actually two neurons here also. We can release the contents of our urinary bladder. With all these motor neurons, with all these efferent neurons, dendrites and cell body are in the central nervous system. Axons and axon terminals are outside. I need to fix what I did here. For interneurons, dendrites, cell body, axon, axon terminal, everything is within the central nervous system. For motor neurons of all kinds, efferent, somatic motor, sympathetic, parasympathetic, dendrites, cell body are in the central nervous system. Axons, axon terminal extend out. That looks better. Our last type of sensory neurons are special senses. Olfactory were just plain bare dendrites. And these are bipolar shape. Let me fix that. These are bipolar shape. So dendrites on one end, cell body, axon, axon terminal. Axon terminal is in the central nervous system. For gustatory, there's actually a special neuroepithelial cell. And that neuroepithelial cell communicates with a bipolar neuron, which then enters the central nervous system. For hearing, there's something called a hair cell. It's got stereocilia, which communicates with a bipolar neuron into the central nervous system. Same thing with head movement. There's a hair cell communicating with a bipolar neuron leading into the central nervous system. And when you see things, you have rods or cones, you have photoreceptors, which communicate with a bipolar neuron, which actually communicates with one more neuron. So we'll describe this later, just like we'll describe the autonomic ones later. So I'm going to draw this one more here, which enters the central nervous system. There's a lot here. I hope you see a pattern though. I show this to give you a reference of every single neuron type that we've discussed so far, and that we'll continue to use in the coming chapters because, yeah, as you can see, there's a lot to discuss with the nervous system, the different things that we sense, how do we decide what to control, and then what do we actually control. We're going to be discussing the brain and cranial nerves, and then spinal cord and spinal nerves. So there's going to be these neurons visible as we go through these chapters. And then we'll wrap up with the autonomic nervous system. So you'll learn why are there 2 neurons in this pathway. And then also for vision, why are there 2 neurons in this pathway, these special cases. So we're going to get practice. An upcoming activity will help you practice this more. I hope this made some sense. Please let me know if you'd like clarification. I'm happy to go over this again. Reach out, leave questions in the comments in the discussion. Thanks, everyone. That wraps up chapter 14. I will see you in the next chapter. Take care.